So my laboratory looks at difficult cancer samples to sequence. So there have been a lot of clinical trials which have relied on using fresh material to attain genomic sequence information, but we have made strides in analysing samples from formalin-fixed paraffin-embedded samples, so your typical histological sample you'd find in a tissue bank. So one of the first things we presented about was how you can access the sequence information of formalin-fixed tissues using FFPE exome sequencing. So we took a series of patients with esophageal and colorectal cancer, and we performed FFPE exome sequencing on them. The way we did it, we adapted an existing Illumina protocol to use some tips and tricks they taught us and some internally developed ones, so that we could use these biopsy samples and accurately sequence their exome to get useful clinical information out of it. In the past, people have said that you can't access these type of samples, that the enrichment from formalin-fixed paraffin-embedded samples won't be any good, but we've shown, and we've talked about in my presentation, that you can use exome sequencing reliably on formalin-fixed material. You can get data that's equivalent to fresh frozen samples, and you can also get data accuracy that's comparable to fresh frozen samples. So this makes these types of samples much more accessible to scientists and clinicians involved in trials research than previously were available. We also talked about using methylation assays, whole genome methylation assays like the Illumina Infinium methylation epic assay. This has been used in clinical trial samples for a while, but one of the problems is it requires quite high DNA input of about 250 to 300 nanograms. In our study, we showed that you can reduce the amount of DNA you put into the sample assay down from anything down to 50 nanograms and still have reproducible quality of assay. So you have the same methylation output as you would with higher DNA output, and you also have the same quality of output. In other words, the methylation calls are the same even with the reducing amounts of DNA. And this is important because methylation is an important marker in disease. It can control gene expression, and in certain tumours like colorectal cancer and glioma, it's important to be able to tell what the methylation status of a tumour is at the gene level, because that will give you information about treatments for the patient. So we've demonstrated and developed methodology that allows you to put very small amounts of DNA into these methylation assays and get a whole genome output that's comparable to the comparison fresh tissue output. So it opens a whole world of clinical trial samples for the methylation assay. Another aspect of the work we've done is looking at circulating tumour DNA. It's a very popular topic in oncology at the moment, learning how we can analyse circulating tumour DNA. All the assays up to this point have basically been based around sequence-based analyses using NGS. So mutation detection in plasma is quite a hot topic at the moment. But as I've said before, methylation is an important genetic marker in cancer, and so we developed the methodology to allow you to do circulating tumour DNA analysis in methylation studies. So we took a cohort of patients with different solid tumours that we'd bled for circulating tumour DNA. We got out small amounts of DNA, as you would expect, anything from 1 to 10 nanograms, and we input this into the methylation assay. And we showed that within good quality control boundaries, you can get a reproducible methylation assay on circulating tumour DNA. And this means that you can follow how the methylome of patients changes over time in the tumour, and will allow us to make decisions based on patients' therapies based on their methylation assay. So it's quite a breakthrough in terms of circulating tumour DNA. And the final thing we talked about was the use of 100,000 Genomes Project data and also our internal amplicon resequencing data to call tumour mutation burden. And as you know, tumour mutation burden is important in that it's a predictor of immunotherapy. At the moment, the markers that we use for predictive response to immunotherapy are things like PD-1 immunohistochemistry and microsatellite instability, but we've shown that with a whole genome and amplicon sequencing experiment, you can call tumour mutation burden to the extent where you can identify patients who may be suitable for immunotherapy. And we've put forward the view that you will be able to cut out things like immunohistochemical testing of MSI and go straight to whole genome sequencing or amplicon sequencing and use this as a marker to decide who needs immunotherapy.